Good morning, Ravi.
Hi, Ramana. It's too early for you, right? 4 o'clock?
Yeah, it's 4:26. got up at 3:45 a.m.
I know. So, yeah, sorry about it. So we have to adjust. Yeah, I mean, for most of the West Coast participants, we need to adjust the timing. So far, I think we didn't have a lot of participants, so there was no demand. But
Yes,
let's see.
yeah, certainly right. Getting up like 4:30 a.m.
Very early. Yeah.
On a weekend is definitely challenging.
Correct. Yeah. Okay, so yeah, let's talk offline with the organizers and try to adjust the time next time.
Yeah. So most of the students are dialing in from home.
Yes, I think everybody is joining from their own cell phone or computer. I mean, scholars, these are all scholars, PhD scholars. They are all coming to department for research work. But now the time, I don't know, maybe, yeah, see, some people are in lab only. I can see Vishnu is in the lab.
Yeah.
Yeah. So since it is only 5 o'clock, probably some students are in the department only.
I see.
Yeah. So now.
Good morning, Ravi Prakash sir.
Good morning, Padmasri. Yeah, I don't see you. Okay, hi, Padmasri. So I see a lot of students, a lot of scholars today.
Yeah, I think after yesterday meet.
Good morning, Venkat. From
good morning. Good morning.
Good morning. I should turn on my video. Yeah, fine. What happened to the video? Yeah, hi.
Hi.
So Ramna Venkat is from GSK. Yeah, Venkat, what year you passed out?
Yeah, 96, 98.
Okay.
Yeah, 96, 98. By the way, my name is also kind of Ramana. My name is a little screwed up, but actually my name is Venkat Ramana. So, but I'll go with the Venkat.
Venkat, okay.
Yeah, my name is Venkat Ramana too, actually. So, but people always call me either Ramana or just by DB, my first, the initials of my first name and my last name.
Okay, great.
Yeah.
Thank you, Ravi, for
Yeah,
for your invitation.
I was searching yesterday on LinkedIn to see who are all chemistry people are out there. So I got hold of a few of them. I think 4 or 5 of them, and sent messages. So Venkat responded quickly. First Venkat name is like Venkat Raman. I thought maybe Tamilian guy studied in Usmania, but later I saw the last name. Last name sounded familiar to me, like a Telugu guy. So we got connected. He lives in Philadelphia area where I live. So nice to connect to you, Venkat.
Yeah, thank you so much, Ravi.
Yeah. Today is our lucky day because a lot of research students are joining. After they had a meeting in department with the HOD, and looks like HOD encouraged all scholars to join. We used to have usually like Yeah, around 40, 50, but I think today we are going to reach 100 or something.
Oh, that's great. Yeah.
Yeah. Usually around 40, 50 we used to get every week. So I think Dr. Padmasri, is it our 8th meeting?
Sir, 8 it is.
8th meeting, yes, sir.
Namaste, Leelavati madam.
Sir, namaste, sir.
Is Arogyam balinattundi?
Cold, sir. Cold.
Cold ainda?
Season.
Acha. Congratulations on
Thank you,
becoming Board of Studies chairman.
Thank you, sir.
Yeah, I think we have some syllabus change meeting scheduled, right?
For PG, we are supposed to change it, sir. That I have to find out.
Yeah. So
UG like implemented new one 3 years before. Recently, it is new one for UG. PG also, I think 5 years we completed. So therefore, we are thinking of going for a change. But the thing is, academic year, I mean, it is delayed and new batch is going to come in a month or two. Whether we are permitted to change at this stage, we have to find out. If so, we'll go for change. Otherwise, by year end.
yeah, I mean, even if the changes cannot be effected immediately, let's work behind the scenes.
We can plan for it.
Yeah, we can plan for it, and please involve I mean, the researchers from US also, probably they will give like Ramna, Venkat, they can contribute what are the latest topics to be added, which is useful for students. So we were talking, Madhavi, me and Padmasri, we are talking to set up some kind of a group where we can discuss and contribute whatever the changes that are important for students. So yeah, I invite all the NRI scientists to be part of it and help the department to make those changes. Yeah.
That's a good idea. Good morning, everyone. I think changing the syllabus more frequently puts us in the cutting edge with the competition with the world, because area every 5 years really expands. For example, I mean, some of you might know in 2000s or 90s, we used to talk about sharpless epoxidation a lot. Then we moved on to something else, click chemistry. Then we moved on to something now very rapidly, the CH activation. So as a result, the area really goes up. And if you don't change the syllabus, we will be really teaching some of those old methods. Of course, we do need to cover the old methods, but they should be very rapidly moved on to new syllabus. That's a good idea, Ravi.
Yeah. So definitely that is one of the things that we should work on. Please introduce yourself, Dr. Ganesh. A lot of new people are there. And your video is
direction is different.
Oh, I see. It's a different direction. I see. Okay, let me put this one.
Yeah.
Ravi.
Introduce.
Good evening, Ravi.
Yeah, Umesh, how are you?
Good.
Thank you for joining on time.
Yeah. Yesterday I conducted a very big meeting for you.
Yes, I saw that. Thank you so much.
Around 150 to 200 scholars attended the meeting, and we asked them to attend every mentoring program regularly.
Thank you. I can also already see the effect. People are joining.
Now they are joining or not?
Yes. Yeah. Now you are there taking attendance, right? So students.
I told them, if you attend regularly, then only I will sign the fellowship form.
Yes. Yeah. I mean, initially
today it will cross 100 or 150, maybe. Hope.
Probably, yeah. Maybe Zoom will not allow after 100 or something. I don't know.
There is a problem.
I think maybe it's up to 300. Padmasri knows
300.
the limit.
Okay. Very good. So
Zoom only for 100 participants, sir. That's why I already started.
YouTube link. YouTube, sir. Further.
You can go to YouTube.
Haripadmasri, madam.
Sir. Sir, it is only for 100. Sir, I've just started the YouTube link also and provided the link in all the groups. Once this is exceeding, they can join through YouTube link and they can engage through the chat section. That's what I
Ganesh
also mentioned in that for the scholars.
Ganesh sir.
Yeah, yeah.
Dr.
Good
Ganesh,
morning.
please introduce.
Thank
Well,
you. Thank you, sir. Thank you.
Good morning, everyone. Yeah, I'm glad to meet you all. I think this program is very good. Designed very well.
Thank you
Yeah,
for accepting our invitation to give the mentoring program today.
Yeah, right.
I am Professor Umesh, Head of the Department of Chemistry.
Nice to meet you all.
Good morning and good evening, everyone.
Dr. Mahendra, good morning.
Good morning. Good morning, Ravi.
Good morning. Good morning, everyone. Good evening.
Good morning. Good morning, madam.
Dr. Prasanna.
Good morning, Prasanna ma'am.
Good morning. Good evening, all. Yeah, it's nice to see that the in-person conference, I mean, the meeting actually, not conference, the meeting with the students, and that's very, very good actually. Once the on-site activities are happening, at least it brings that more momentum for the students and
So, sir, shall I start the program?
yeah, Padmashri, I think you can start. I mean, we're already past 7 minutes, right? I think we can start the program.
Yes, I think we should go.
Yes, sir.
Yes.
Hello, madam.
Yes, sir.
Admitted నాకు వస్తుంది కొందరి messages.
Sir, you have joined through the department one, so probably it is asking you.
And I should do for everybody.
You click admit, sir, because we both joined from the same.
Yeah.
Admit, sir. One second, I'll just start.
Sir,
I'm starting. Can I, sir? Head, sir.
Hello.
Sir, Umesh, sir, I'm just starting the program. Can I?
Yeah. Okay, madam. You can start. Start the program.
Okay.
Start saying, madam.
Yes, sir. Good evening, everyone. Welcome all the scholars, newly joined scholars, the faculty members and all the mentors, and today's speaker or mentor, Dr. Ramana Garu, and all the other mentors. Welcome you all on behalf of the department HOD for this meeting. Welcome, Professor Leelavati ma'am. Congratulations on all our behalf for taking up as the chairperson BOS of the Department of Chemistry.
Thank you.
Simultaneously, I'm just clicking admit all the time.
Even it's coming for me also. I am also clicking.
Yeah, you are the co-host, no, ma'am. That's why.
Yeah, even
if I don't
clicking.
click, it is coming to you.
Yeah, even I'm clicking.
So now I formally welcome Professor Leelavati ma'am.
I think the audio went silent.
Yeah, Dr. Padmasree. Are you with us?
All?
Yeah.
Yeah, now it's better. It's better. We can hear you, Dr. Padmasree. Yeah, we can hear you. Your voice.
అదేనా అదే కనబడతలేదు.
Padmasree, she stopped her screen sharing and then
Maybe if we all cut our videos, maybe audio might work very well. Looks like either we lost her or are we not able to hear.
Now she's back.
I think we can, sorry, can I take over until she comes back? Good morning, everyone. Can I request Professor Leelavati Madam to give her formal welcome address, please? Madam?
yeah, right, sir.
Yeah.
So I thank Padmasree for that introducing me. So Professor Umesh Kumar, Head Department of Chemistry. Are you able to hear me, sir?
Yes, yes.
So Professor Umesh Kumar, Head Department of Chemistry, Dr. Haripadmasree, Coordinator for this program, and Dr. Ravi Prakash Nereddy, Dr. Madhvi, Dr. Prasanna, Dr. Mahendra Diwal, and other mentors of OU Chemistry alumni, and Dr. Ramana Doppalapudi, today's speaker, dear colleagues and research scholars. Hello, everyone. With great pleasure, I extend a warm welcome to you all on behalf of the Department of Chemistry for this Step-Up Mentoring Program 8, organized by the Department of Chemistry in association with Alumni Mentors Group, Usmania University. It's indeed my privilege to welcome the eminent members from the chemistry alumni who are a great asset to the department. As you all are aware, we have completed almost 7 sessions with a break in between because of the examinations of the students, and today is being the 8th one. So in the previous sessions, we heard mentors like Mahendra Diwal, Madhvi, Prasanna, Dr. H.S.P. Rao, Dr. Bhaskar, etc., who shared their experiences with our scholars. So in these programs, students will have the chance to learn from those who came before them and continued on in certain careers and educational paths. So one of the main objectives of this program is to provide the opportunity for students to obtain guidance on career goals and to build academic, social, and professional networks. Especially in this pandemic, with students in remote learning, our alumni are even more crucial to the success of the students. So hence we request all the research scholars to actively participate and make the most out of it. So I'm sure definitely, and combining with this, with the yesterday's address by our head department of chemistry, the students are definitely motivated, and we expect large numbers from today onwards. So on a final note, we are very grateful to OU Alumni Mentors Group for sharing their professional and personal experience and expertise with our students. So thank you all. And I request Dr. Ramana Duppalapudi, sir, to take up the session. Thank you.
Yeah, a quick bio. Thank you, Professor Leelavati Madam, for the welcome address. A quick bio about Ramana, a good friend of mine. Ramana is currently Vice President, Avidity Biosciences, California. Dr. Duppalapudi, a native of Khammam district in Telangana, received BSc and MSc Organic Chemistry from our proud Usmania University. He has completed his PhD in Organic Chemistry from IICT, Hyderabad, then conducted postdoctoral research at UT Southwestern Medical Center and Southern Methodist University, Dallas, before moving to San Diego, California, in the year of 2000 for a career in the pharma and biotech industry. For the last 21 years in the pharma and biotech industry, Dr. Duppalapudi developed multiple clinical candidates for the treatment of cancer, muscular diseases, and wet AMD. He currently lives in San Diego with his wife and 2 children. Dr. Duppalapudi loves to play tennis and a regular hiker of local mountains. I welcome Ramana. It is a great honor to have you here and interact with our students and scholars. In terms of his research, he's a scientist with experience in the discovery and development of variety of drugs, including peptides, antibodies, oligonucleotides, and antibody drug conjugates. So that's his research interest. Yeah, please go ahead, Ramana. All yours.
Thank you, Ravi. So can I share my slides?
Yes, Padma, I think she has to stop the screen sharing.
No, I think Ramana sir can directly upload screen share. I think you are already host, sir, co-host.
There is a share screen at the bottom, so you can click on it, I think. Let me see if I can. Okay, yeah, good.
What to do today is to give you a very high-level view of nucleotide therapy. Some because somewhat recently developed therapeutics. Give me 1 second and let me. So, screen now?
Yes, it's perfect, sir.
Yes, it's good now. Please.
So, therapeutic tools and approaches, right? There are several of those. I am giving you basically took a picture from the British Journal of Pharmacology to give you a very high level view. If you start from the left side bottom, the small molecule drugs, right? These are all the drugs that we see, the pills that we take, and mostly the oral medications. These are really small in size, and they act on a variety of targets. And then more recently, in the last 3 to 4 decades, came up the protein therapeutics, or what they call them as biologics. These are the antibodies or small proteins or hormones. They tend to be, usually in liquids or lyophilized powders. You need to dissolve them in some sort of solution and give, administered to the patients in the form of IV or subcutaneous, injections. And if you go to the top, and I'm sure many of you heard, gene therapy. This is coming up in the last couple of decades with a lot of successes and also failures. Today, I would like to focus on a few things. These are the siRNA therapeutics and then also the antisense oligonucleotides, and give you a little more details in the next few slides. And then the other things are also the real recent gene editing technologies. That means you can go in and correct or edit the genes of the mutated genes or the incorrect genes to express the right protein. And CRISPR-Cas9 technology is one of those you probably have seen quite a bit in the news and also in the in the scientific journals. And the last but not least is the cell-based therapies, where these are you can take the cells from the patient and then treat them with to express certain proteins, or essentially modify them and then introduce them back into the patient. And if you look at the properties of all these different types of therapeutics, their properties actually depend on their type. If you look at the small molecule versus the antibody therapeutic versus the oligonucleotide, the molecular weight increases. The small molecules are typically, they're about 500 Daltons, or some of them are even maybe they go up to 1,000 or 1,500. And in the middle, you have the antibody therapeutics. These are the really large. Some of the for the full length antibody therapeutics are about 150,000 Daltons. And the oligonucleotides are typically around 7,000 to 14,000 Daltons. As the size increases, right, it becomes really difficult for them to get into the cells. That's why if you look at the cell permeability, the small molecules actually, they get into the cells, the tissues very easily, but not the large molecules. Same thing goes with the oral bioavailability as well. That means if you take these drugs orally, small molecules can go into a digestive system. From there, they can get absorbed and get into the bloodstream and then go to the different tissues from there. Whereas antibody therapeutics or protein therapeutics or oligonucleotides, when you take them orally, essentially they get digested in our digestive system, and can you hear us okay? Can you hear me okay?
Yes.
Yeah.
You can hear okay.
Yeah, please go ahead. You can hear you fine.
And then the site of action, right? Because the small molecules are cell permeable, they can actually work on they can go after the cell surface targets and then also intracellular targets, because the cell permeability of these molecules are pretty high. Whereas for antibody therapeutics, it is usually, they can only go after the receptors expressed on the surface of a cell. And for the oligonucleotides, it is actually most of their targets are intracellular. Certain aptamers, they can actually bind to proteins on the cell surface, but their mode of action is primarily intracellular because they act on the essentially on mRNA or DNA. And then the bottom, as I have seen, primarily the small molecules are oral, and antibodies and oligonucleotides are primarily IV or subcutaneous dosing. And this in the next slide, you might ask, what are these oligonucleotide therapeutics? As you know, the central dogma of the protein expression, we all have genes, that is the DNA or the deoxyribonucleic acid, and then it is transcribed into, it transcribes the messenger RNA, and which then translates into a protein. And I'm showing here, give me 1 second. Can you guys see the laser pointer?
Yes, sir.
So essentially the DNA is, it's basically a sequence of, nucleotides. Each nucleotide contains a base unit and then an internucleotide linkage, the phosphodiester. The DNA contains essentially, there is no substitution at the 2' position on the sugar. Whereas in the RNA, it's the same structure, but you have a hydroxyl group on the 2' position. That's why the DNA is called as deoxyribonucleic acid, and then RNA called as simply ribonucleic acid. And then, so as you know, we all kind of, the human genome has been sequenced. Now we kind of understand, what different genes in our genes, and then what genes are responsible for expression of, all of the proteins in our body. So that means, in case if you want to develop an oligonucleotide therapeutics, it should be fairly straightforward, because you have the mechanisms. Sorry, I think I missed something, and just let me take one step back here. And then most of the small and large molecule drugs are either activate or deactivate at the proteins. Of course there are always some exceptions, but if you take a disease, maybe a protein is its expression is lacking, or it has too much expression, you have, causing the disease. These small molecules or these protein therapeutics, large molecules, they work primarily at the protein level. And the oligonucleotides, on the other hand, they go one step prior. That means they actually go and act on the genes directly, either on the DNA or on the mRNA. And then the result is, they change the expression of the protein that is causing the disease. It could be in some cases, they lower the expression of the protein, and it could be in some cases they correct, the misfolding of the protein. So, but primarily that's the main difference I want you to understand. The difference between the pills that we take versus these new class of drugs, the oligonucleotides. And how exactly do they work, right? There are many different types of, once again, these gene therapies, but I'm just focusing on a few. And so if you look at on the left side of this slide, it says ASO, and then it shows like, a picture of the ASO. This is essentially, antisense oligonucleotide. And what you're seeing is a single strand of a DNA. It's a small DNA. Usually it's about 15 or 16 mer. And then what happens is, when we dose these patients with this antisense oligonucleotide, it goes into the cells and then essentially binds the target mRNA. As you know, the DNA, RNA, they form the complementary sequence. That means, a sequence, depending on the complement, the sequence that is complement, to based on the therapeutic, it goes and finds the right molecule, the right mRNA, binds it, and then either it acts as a steric block. That means, it now essentially makes this molecule of mRNA inactive, because, and then in some cases it actually recruits certain protein factors like RNase H, and then results in the decomposition or cleavage of the mRNA. Once again, that results in the lowering of the protein expression. In some cases, it actually modulates, it actually binds the pre-mRNA and then modulates the splicing of the, because when the DNA is translated into mRNA, there are certain sections that are removed from the DNA before it is converted into mRNA. That process is called splicing. And these molecules will go in and then change the splicing to change the function, to help express the functional protein. And then there are a lot of approved drugs lately, Mipomersin, there are a bunch of those. You may have seen in the newspapers lately that, at least I have seen in some of the Telugu newspapers, that a small child with the spinal muscular atrophy, the drug.
We can't hear you now.
Yeah. So, and I was saying the cost of these drugs, and then this is about the antisense oligonucleotide therapeutics. On the right side of the screen, you have siRNA. These are the short interfering ribonucleic acid molecules. And the difference between the antisense oligonucleotide and siRNA is, now this is a double.
We can't hear you. Ramana, we can't hear you.
Yeah, apparently the Zoom is not friendly anymore, like before.
Yeah, it's weird.
It's weird. Yeah, very weird. I don't know.
Sir, excuse me, sir.
Yeah, Dr. Ramana, we can't.
We can hear you. Yeah. Hold on a second. Let's figure this out.
So the host is me, actually.
Yeah, I think we can hear you now. Now we can. No, again, it's gone. The voice is gone.
Also, there is some background noise somewhere. Anybody?
I think there is a problem. We can't hear speaker's voice. No, yeah. Dr. Ramana, we can't hear your voice at all. Dr. Padmashri Garu, are you there? Yeah, we can't hear you, Dr. Padmashri.
Sir, otherwise rejoin the session, sir. Exit and rejoin the session. If any connectivity issue will be rectified.
Oh, okay. Can we do that? Ravi Prakash Garu.
And one more thing, sir, my suggestion, stop recording, sir. Anyway, it is YouTube line. We are in India with very poor bandwidth, sir.
Oh, I see. That could be a problem.
We are also having issue with the network. Recording along with live means, I think our little bit problem.
No, we didn't
yeah.
Problem. It was in other session. Only today, I think.
ఇది వస్తున్నట్టుంది సార్, అందుకే ఇది ఒక రావట్లేదు. మీరు పర్సనల్ దాని నుంచి జాయిన్ అవుతారా? ఎందుకు సడన్లీ ఆడియో పోతుంది?
ఎవరు అందరు పర్సనల్ దాని నుంచి ఎవరు జాయిన్ అవ్వరు?
నాది రావట్లేదు ఎప్పటి నుంచో.
I think now we can hear you. Let's continue. Let's see. Dr. Ramana garu, keep going. No, we can't hear you. That's the problem. It's weird. Can you rejoin, Dr. Ramana garu? Yeah, I think that would be better.
Please, all are requested to mute yourself. So much background noise is there.
ఆల్రెడీ అడ్మిట్ చేసినాం కదా వాళ్ళు ఏం వెళ్ళరు.
మ్యూట్ చేసుకోమని చెప్తున్నాను.
Yeah, whoever is not speaking, please mute yourself while you're not speaking, please. Padmashri garu, మీరు కూడా mute చేయండి, I think, when you're not speaking. Yeah, you're muted, Dr. Ramana. Unmute yourself.
Okay.
Yeah, now we can hear you.
Yeah, what was happening was I could not unmute myself. So
please go ahead, share your screen and continue.
Yeah, let me go back to the share slides. So host disabled participant screen sharing. So whoever is hosting me, please give me that permission.
Padmashri garu, let me see. I can give you.
Yeah, now you can do, sir.
Okay. I still can't do that.
Okay, let me see if I can help you on that. Yeah, try now. Ramana, try. Yeah, you're muted.
Okay, yeah, can you see the screen?
Yeah, we can see the screen, your screen. Yeah, go ahead. Keep going, please.
Okay. Okay, so we spoke about the antisense oligonucleotides. Now getting back to the duplex siRNA or short interfering RNA. When I say duplex, that means there are two different strands in this one, both sense and the antisense strand in this. That's the main difference between these two, or one of the main differences between these two types of therapeutics, and also the mechanism of action. And so the siRNA oligonucleotide, basically once it gets into the cell, it interacts with the AGO2 protein and then forms a complex called RISC, the RNA-induced silencing complex with the target mRNA, and then that leads to the cleavage of the mRNA. And then going back to the dogma, right? When we target the mRNA with these two therapeutics, essentially what you're doing is changing the expression of the protein that is causing the disease. And in both cases, whether it is antisense oligonucleotide or siRNA, there are a lot of therapeutics approved recently. And I think in RNA there are two drugs that are approved, and then the third one called inclisiran is getting very close to the approval. I'll talk about that towards the end of this presentation. If you look at the typical structure of an oligonucleotide, right? This is, I think I mentioned this a little while ago, and I'll talk about it one more time here. This is essentially the structure of a single strand of a deoxyribonucleic acid. You see the base, the sugar, and then the phosphodiester linkage, right? So there are, In the DNA, there are 4 bases, cytosine, thymine, adenine, and guanine. And then, the guanine always forms, the hydrogen bonds with the cytosine, and then, adenine forms with the thymine. On the RNA, you have basically the cytosine, adenine, and guanine. The thymine is replaced by uracil. So, and then the RNA, the difference is also at the 2' position. That means right here you have a hydroxyl group. That's the main difference between the structure. That's the structure of an essential oligonucleotide. So, just going back, right? So, as I mentioned earlier, right, we know the structure or the sequence of the DNA, and then also the mRNA that is causing the disease. And then we know that the mechanisms that actually inhibit these molecules, and then it should be fairly easy to come up with therapeutics, right? Yes, it is actually very easy to come up with therapeutics. And then it's also fairly easy to make them. I'm sure you've seen some of these, the oligonucleotides, synthesizers, either at the Spania University or elsewhere. These are fairly easy to make. You start with the solid phase synthesis, where you have one of the sugar loaded onto the solid phase, and then go through a sequence of steps. The removal of the protection groups, and then adding the next nucleotide in the form of an amidite, and then doing the oxidation step, so that the cycle continues. This is basically an automated synthesizer. And essentially, you see the different, solvents. This ports for different solvents, and also ports for different columns and reagents and stuff here. And then this is like an acta pure. This is sort of like an HPLC that you have in the lab. And these are the ones you can once you grow the length of the the oligonucleotide on the solid phase, you can cleave it and purify it on these instruments, and then analyze them once again on the LC-MS. This is the HPLC attached to a mass spec instrument to give you the the purity and also the identity of the final oligonucleotide that is made. So if I just take back a minute, right, so we know, as I mentioned before, we know the structure of the genes, we know the mechanisms, and we know, how easy it is to make the oligonucleotides. And you might ask why there aren't many drugs or oligonucleotide drugs on the market, right? It turns out actually developing an oligonucleotide therapeutics is really challenging. Part of the reason is, the oligonucleotides in the form they're very rapidly degraded. That means if you go back and, to this structure, this internucleotide linkage is not, with this phosphodiester linkage is not very stable in circulation, or once it gets into the cell, nucleases actually rapidly, and other enzymes rapidly hydrolyze this molecule and then cleave this, and cleave the oligonucleotide. And then also, they're hydrophilic in nature. That means these are all water-loving molecules. Once again, coming back to the phosphate, this is highly charged usually, and these molecules are very hydrophilic. That means they're highly soluble in water, and they do not get through the hydrophobic cell membrane, so they do not get into the cells. And the third important thing is, these oligonucleotides are actually very rapidly cleared from circulation. That means once we dose it, that our body tends to, eliminate them from the circulation through the filtration at the kidney. And then, I'll get to the last 3 points in a little while. But for with respect to these problems, right? These 3 problems I highlighted here. Chemists always come to the rescue. Chemists are, in a way, engineers. How engineers build those big buildings, and chemists actually construct molecules. We look at the molecule. Okay, oligonucleotides have a lot of promise, but at the same time, they're really difficult to convert them into therapeutics. what are the problems? The first thing is, oh, the internucleotide phosphodiester linkage is very unstable. It hydrolyzes very rapidly. Then chemists get in and then they look at, okay, how can I make that change or make it more stable in circulation? That's how they came up with this phosphorothioate linkage. If you remember the other slide, the internucleotide linkage, basically where you have the sulfur here, you have an oxygen there. But when you convert this into a sulfur, it becomes, instead of a phosphodiester, it now is a phosphorothioate. And this is highly stable in circulation, and then also stable to the nucleases. And then, but this one, right, you're not really changing much in the molecule. You're just changing, replacing oxygen into sulfur. That changes everything in terms of stability. And there are some other chemistries where people looked into, this is basically now the 5-membered sugar as being changed to a 6-membered morpholino ring. The rest of the nucleoside part doesn't really change much. The base remains the same. The 5' hydroxyl remains the same. But instead of the phosphodiester, now they actually change that into phosphorodiamidate linkage. This is also nuclease resistant, highly stable molecule. And then the more in advanced therapeutics are more further modifications is the peptide nucleic acid, where they change the backbone completely. Now you are moving away from these sugar moieties and are replacing them with amino acids, but they retain certain structural elements of the DNA or RNA, but they're highly nuclease resistant. They bind to their complement sequence, with a very high affinity as well. And then there are other solutions that chemists have proposed. These three basically focus on the internucleotide linkage, right? But other nucleus where people looked into what else can I change, right? There are a lot of modifications that chemists have played with, but I'm focusing just a few. One of the key structural changes, the modifications, is the 2' position on the sugar moiety. You have, this is where the base is attached to the ribose. This is called position 1, and then position 2, 3, 4, and then this carbon right here, is the position number 5. The 2' position, chemists really played with it quite a bit. And if you convert that hydroxy into a methoxy position, then it changes the target affinity. It actually provides some nuclease resistance, and it's also very well tolerated in many modifications. And then very similarly, you also have the 2' mo, or this is the methoxy ethoxy, is the more modification, right? And then there are other modifications that are also very prevalent. This is a locked nucleic acid, where you basically lock using a methylene bridge, lock the 2' hydroxyl with that of the 4' position. And all this basically improve the binding affinity of the resultant oligonucleotide, and also they increase the activity of the oligonucleotides. And then this is one of the structures of the mupamycin. This is an antisense oligonucleotide. You can see the sequence here. And you have a gapmer. This is basically a DNA, and as you can see here, this is a phosphorothioate. That means this structure here, because as I mentioned, phosphodiester hydrolyzes rapidly phosphorothioate and provides a stability. And then the more modifications on both sides, the 5' wing and the 3' wing, provides more stability as well. This is one of those approved drug that is now currently being used to treat patients. And then there are other companies as well. Alnylam Pharmaceuticals, this is actually a leader in the siRNA therapeutics. They use even more other modifications. You can see each color, in each circle is a nucleotide. And then each color of that nucleotide represents certain modification. It could be a 2' fluoro or UNA. This is unlocked nucleic acid. GNA are many different modifications that each company came up with their own way of their own way of their own pattern of chemical modifications. I just want to highlight one thing about Alnylam Pharmaceuticals. This company is actually leaders in the siRNA therapeutics, and the chemistry. The head of chemistry is Dr. Muttai Manoharan, and people call him as Mano. And he is one of the best oligonucleotide chemists in the world. And and he is and he has given I'll talk about his contributions, but he is in a way, right, single-handedly changed the siRNA therapeutic field. And then there are other companies as well, Arrowhead, Silence Therapeutics, Dicerna. They all use different chemical modification. But what I want to say, right, when there are challenges, the chemists are the ones that come up with solutions with respect to these molecules. That's the role of the chemistry I see in the industry. Look at the problem. How do I make chemical changes to these molecules so that they become better drugs? So we can now increase the stability and activity, but as I mentioned before, right now, these oligonucleotides, they do not get into the cells, right? But how do we get them into the cells? That's where the targeting point is. That means that's where you take the help of the ligands that bind to the cell surface receptors. And the structure that I'm showing here is a GalNAc siRNA. GalNAc is a sugar moiety, three different, the sugar units combined. This is called tri-GalNAc. It binds the acyloglycoprotein receptor, or ASGPR, on the liver. This molecule binds it, and then it gets internalized. So now, by using this type of mechanism, you are solving the problem of getting these oligonucleotides into the cell. Once it gets into the cell, it gets out of the the endolysosomal pathway and then gets into the cytoplasm, where it can participate in the mechanism. And that's the RNA inhibition or mRNA inhibition. The receptor, on the other hand, it gets back to the cell surface by the receptor recycling. So so if I just want to make the point again, now you are basically taking help of another molecule to increase the the internalization or intracellular delivery of this otherwise difficult to otherwise challenging oligonucleotide therapeutics. And this is the identifying this particular GalNAc moiety, and this technology was the major contribution by Alnylam, and then it was actually developed by Muttai Manoharan at that company. And that was his contribution. Today, if I remember the market capitalization of Alnylam correctly, this is, I think they're pretty close to $10 billion. And it is primarily, most of the valuation is coming from the identification of this particular technology and the products they developed by using this technology. So by using these GalNAc technology, right? We can. There is one more slide here. This is typically the structure This is the siRNA, the oligonucleotide, and then this is the GalNAc, the way they're constructed by Alnylam Pharmaceuticals. And they certainly help the delivery of oligonucleotides to liver, right? But this is the key, what is the key point here? The GalNAc only helps the oligonucleotide delivery to the liver because its target receptor is expressed in the liver. But what about the other tissues? If you talk about heart, muscle tissue, or some other internal organs, right? And these are called, in the oligonucleotide non-hepatic tissues, right? And how do you do? What is the solution for it? There's so many companies are working on different other technologies. And if you start with A here, this is a picture of an image of an antibody therapeutic, and then the green lines here are the oligonucleotides. Essentially, you conjugate, just like the way you're conjugating the oligonucleotide to a GalNAc. Now you're conjugating this to the antibody. I'll get to this in a little bit. And then there are other technologies where you conjugate this to a long chain polymer. And then, also certain, so the targeting moiety is attached to it. And this is, we just spoke about, the GalNAc conjugates. And then the D here is the lipid nanoparticles. And here you take, a lipid bilayer, and then inside the lipid bilayer based nanoparticle, you have the oligonucleotide. When you dose these, these can go to different tissues, and then because of their nature, they get, as a particle, they get into the cell, and then once they get into the cell, they disintegrate, releasing the oligonucleotide. Similar thing, the E here is the gold nanoparticle. You take a tiny nanoparticle of gold, and then decorate it with the different oligonucleotides, and then now it has you're overcoming the kidney clearance barrier, and hopefully it gets into different tissues, gets as a part of the IV dosing, and then slowly gets into the tissues to deliver the oligonucleotides. And then there are other mechanisms as well. What I am working right now, our company working on this particular technology, the antibody oligonucleotide conjugate technology. And I'll show you, what we are doing, at a very high level. And then, as I said, this is the, we are primarily working on the siRNA, or the short interfering RNA. And then, just to tell you about the antibodies, right? These are really large, 150,000 Dalton protein therapeutics. And they, once you dose them, the human or humanized antibody in humans typically has a half-life of, 3 weeks. That means they survive in the circulation for a long time. And then they bind their target receptor with a very high affinity. And once they bind it, depending on the receptor and the antibody, the binding affinity and the receptor properties, they internalize into that particular cell. So in that process, whatever is attached to the antibody, it takes that molecule into the cell. So we are depending on that particular mechanism. And so essentially the role of the antibody is to, when you conjugate an oligonucleotide and antibody, the role of the antibody is to take that oligonucleotide to the tissues where antibodies, the target receptor is being expressed. And then increase the, change the distribution of the oligonucleotide, and then also the pharmacokinetics. You probably cannot see it very well on this particular screen. What I'm showing here is on the x-axis, this is the time, and the y-axis is the concentration of the injected oligonucleotide in two different forms. One is the unconjugated oligonucleotide. You can see the green one, it actually rapidly cleared from the blood. Basically, within minutes, it is gone from the blood. Whereas When you conjugate an antibody, it then remains in the circulation for a long time. And as since the antibody therapeutics have been around and that they're manufactured for a long time, and there are a lot of technologies available, and there, the manufacture of these drugs is not that difficult, even though it's challenging, but not that difficult. And it provides, because of the wide variety of the receptors, these antibodies, cell surface receptors, these antibodies can bind. It expands the therapeutic opportunity. And then on top of it, it also allows, the molecular biology has improved so much that, you can actually engineer these antibodies and then, optimize them to the needs of that particular disease. And so essentially we are combining the specificity of the siRNA based therapies with the selectivity of the antibodies and improving the delivery of oligonucleotides to non-hepatic tissues. And that's our technology. And before I conclude the lecture, I just want to show you the power of the oligonucleotide therapeutics. This is a recently, it is not approved yet, but it will be in the next few months. A drug called inclisiran. This is an siRNA therapeutic. It targets PCSK9, resulting in the low density lipoprotein C reduction. What you see here is the LDL, and the base levels before the treatment, initiation of the treatment, they're about 130 milligrams per ml. But after the treatment, eventually it goes down to about 60, or total is about 55% reduction. You can achieve this as well. Essentially, this is a sort of cholesterol reduction, right? You can take some of your family members, maybe taking the cholesterol drugs like Lipitor, and these are sometimes you need to take them once a day, some of them 2 times a day, pills, right? But there are a lot of side effects, the liver issues with them. But one needs to take them, once or twice a day. But look at this particular drug. You basically give, a initial dose and then a loading dose after 3 months. After that, it's actually in one injection every 6 months or 180 days, right? And then this is basically 3 months, 6 months, 9 months, another 6 months, another 6 months, right? In a sense, and it keeps the LDL-C levels constantly low, are reduced by 55%, and that's the remarkable nature of the other power of these oligonucleotide therapeutics. That the inclisiran is incredibly safe molecule if you look at the clinical data, compared to the other PCSK9 inhibitors, like there are few antibodies that are also approved. And this was actually developed by Alnylam, but they licensed it to a company called The Medicines Company, and Novartis actually bought the Medicines Company just for this drug for, I think, little more than $10 billion. You can convert that into Indian currency and then see the value. So there are not that many companies, I think, doing research on the oligonucleotide therapeutics, but this is it's been around for a long time. There are a lot of disappointments, but I think eventually, right, the chemists have solved the many issues in this problem, and then they are now delivering on their promise. There are more than a dozen therapeutics approved by FDA, and a lot of chemical solutions are being are found to improve the stability And then, as I mentioned, the duration of action is the hallmark of these therapeutics. And then the delivery to the liver has been in a way changed, solved by these lipid nanoparticles or the GalNAc conjugates. What is next is the delivering to the non-hepatic or outside of the liver to other tissues is the hot research topic. That's where Avidity, my companies, or our company is working. And then there are a few other challenges as well. Once these oligonucleotides get into the cell, they do not get they get into the cytoplasm. And, so we are also working on those. So last two items are really hot research topics. And then once we can solve those, you will see this whole area exploding into several therapeutic areas. We may actually see, several other drugs that can, essentially, instead of, daily dosing, we go into this once every, 6 months or maybe once a year type of injections, not too, far into the future. And that's the last slide of my presentation. And hope I was able to give you some useful information to all of you.
All right. Thanks, Dr. Ramana. It's a wonderful presentation, and we invite discussion on this. We have eminent scientists here, Dr. Ganesh Tota, Dr. Mahendra from US. Praveen is also here. So yeah, let's initiate the discussion, and also students, please, if you have any questions, please ask the questions. Yes. Anyone?
So definitely, this is Ganesh. The oligonucleotides apparently offer very advantages compared to small molecule drugs or drug-like molecule. In simplistic term, what advantages do they provide? What side effects will they not have compared to small molecules? Dr. Ramana, if you could.
Yeah, so as I mentioned, right, the major advantage with the oligonucleotide therapeutics is the duration of action, right? They can, once you identify the right therapeutic, the frequency of dosing will change. And then the number two, right, is, the oligonucleotide therapeutics open up new avenues for drug discovery. For example, right, the small molecules, they're primarily they target the certain enzymes intracellularly or certain cell surface receptors. And when it comes to protein-protein interaction, reducing the protein-protein interactions, small molecules actually do not succeed very well. That's where the large molecules come in. But with the oligonucleotides, essentially the duration of action, and then also opening up the new targets, and then also the specificity. I think you asked about the side effects, right? So oligonucleotides are highly specific. And then when it comes to side effects, there are two different things, right? One is the on-target toxicity. That means, by reducing or changing or modulating the target protein, that can, it will have some benefit, but it may also have some side effects, right? But off-target side effects, where these are unintended, these can be reduced by the oligonucleotide therapeutics because they are so specific.
Yeah, thank you. So the long duration of action, is it due to the stability of these oligonucleotides, or it's the actual action they bring into the biological systems that make them to act very longer time?
Yeah, I think that's a very good question. These, you basically gave the answer, So number one, yes, as all these chemical modifications made these oligonucleotides highly stable for a long time. That's one thing. The second thing is, if you look at the siRNA therapeutics, right, the risk the risk means RISC, the RNA-induced silencing complex they form. Okay, thank you. So the RNA-induced silence, that actually once the antisense loads into the RNA silencing complex, induced silencing complex, it stays there for a long time and then essentially chops down. That leads to the reduction of the mRNA.
Okay. Yeah, this is Mahendra Bidwal.
Hello, sir. This is.
Yeah, go ahead.
Hello. Good evening, sir. Sir, regarding that cholesterol medicine, who is taking no, sir? Instead of that injections, you told no, sir. Now, actually, what is the usage of using instead of that medicine while taking of injection? What is the difference between those two, sir?
Difference between taking, for example, a pill versus the injection. So is that the question for, I think it is from Farveen, right?
I think that's what is the question.
Yes.
Okay. So it is basically, right, do you want to take, a pill or, once or twice every day, yes, or do you want to take an injection like once every 6 months, right? So that's the major difference, right? Some people, they may prefer taking a pill, or two every day, but some may prefer, I just want to take one injection every 6 months and then I forget about taking these medications every day. So I think there is one thing is conveyance, right? The second thing is, because of the specificity, I think that's what Dr. Ganesh was asking, a little while ago, specificity of oligonucleotide therapeutics, the side effects that you get from them are fewer. If you look at least, my parents take Lipitor, my uncles take Lipitor in India, and none of them are, I mean, of course, it certainly provides benefit, but there are a lot of side effects. And my uncle is actually forced to go off the Lipitor medication, once every few months because of the liver toxicity he sees. He develops jaundice. That is primarily driven by him taking these cholesterol lowering small molecule pills.
Okay, thank you, sir, for giving information.
Yeah, this is Mahendra Bidwal. Dr. Ramana Garu. It's an excellent overview of nucleotide chemistry. You not only gave the entire, the review of the field, but also you emphasized the role of a chemist's role in influencing the biology with simple modifications to the chemistry. It's an excellent overview. I have two questions. And my first question is kind of a more oriented towards your company. You said you're modifying the antibodies. You're conjugating the antibodies to the oligonucleotides or siRNAs, what you're developing in your company, right? My question here is, what kind of modifications are you guys using? Number one. Number two is, are you using the whole antibodies only, or are you using any antibody fragments or nanobodies or any single chain variables, things like that? That's my first question. And once you answer this, and then go to the second question.
Okay. So, Mahendra, we are actually the modifications on the antibody that they're asking. So antibodies are complex large molecules. As you know, they they activate the FC and the bind FC gamma receptor, and then lead to some effector functions. And that's one of the things, the therapeutics that we are developing will require essentially nullifying the effector function. So we made some modifications to the heavy chain of the antibody to reduce the effector function. And then also we made some changes to improve the binding affinity of the antibody therapy and of our antibody. So that means we are the modifications that we did allow us to very quickly identify the antibody that is optimal for the oligonucleotide delivery. And we actually tried fragments of antibody like Fab or Fab2, and we haven't tried nanobodies, but we tried antibody fragments and full-length antibody, and finally settled with the full-length antibody primarily because of the long pharmacokinetics that full-length antibodies provide. Because of their FcRn mediated recycling, they have long half-lives.
Awesome, thank you. The second question is, so you have spoken about the modified nucleotides, right? To make them more stable, you're changing you're doing small changes in the phosphodiester linkage, right? So in general, the mutations in the DNA or genome happens to the modifications to the nucleotides in general, to be say like DNA methylation, DNA acylation, or some of the major causes of the mutations in the DNA, right? What are the consequences or what are the chances of these modified nucleotides when you're delivering to the body? They're going to go into the human genome and then imbibe into the genome and then modify our DNA. What are the chances and what are the consequences? How can you predict this, and how can you stop this? Can you please shed some light on this?
Yeah, so this is our bodies, right? They do actually recycle some of these nucleosides, right? Basically you take a nucleotide, body hydrolyzes them and picks up some of these. But it's an enzymatic mechanism, and I don't have a great deal of knowledge how exactly it happens, but that's where chemists made a lot of modifications, right? Many of them did not survive precisely for that reason, right? Some of the ability of our body to incorporate into our own genes, right? People have looked into it; those were quickly eliminated. The ones that I showed are the ones that survived. They do not, or actually they have minimal incorporation into the genome. If basically I think the mechanism is, the nucleoside forms and the nucleoside triphosphate that's the one, triphosphate is the one that gets into, enzymatically, it gets into the growing, the nucleic acid chain. And these substitutions, the one that survived, the 2'-methoxy or MO or LNase, they have a much smaller tendency to get incorporated into the host's genome.
Thank you.
Hey, Dr. Ramna, this is Satish. I have a couple of questions. I'm in a classical drug-based, structure-based drug design this last 25 years, and this oligonucleotide chemistry is about maybe about 10 years back, about 30 years back, first introduced, and from then a lot of changes happened. I'm sorry I joined the talk a little late. I did not start the very first. Maybe you might have already addressed this question, but there are a lot of challenges compared to classical drug design, drug discovery compared to in oligonucleotides. So how far we came in this thing, overcoming problems, and are there any still challenges? Drug discovery, not identifying is different, but how you go in the sequence of identifying a target, and how you modify your your molecule, like not like a regular drug discovery. How you came up? I mean, how far we came in identifying and changing your work, how to go after a treatment. And the second thing I noticed is when you inject, it's not like injecting in the shoulder, right? More of treatment the patient. Can you shed a light on that one?
Satish, are you from ISCT? You are?
Yeah, I'm here junior. I'm Dr. Ramara.
Oh, okay. Yeah. So it's been a long time since I haven't seen you. Why didn't you come on the camera for a second?
I'm sitting in the dark.
That's okay. I think these are good questions, Satish. You're asking, if I understand your first question correctly, right? The small molecule chemistry, the optimization these days, it involves a lot of structure-based drug design, right? You take your lead molecule and then you try to fit that into its target, binding moiety of its target protein. And then, you use the computational chemistry to make further changes to the small molecule, right? So changing a few things here and there, and then eventually end up optimizing it as a drug. And that kind of thing, right, is not very well developed. Maybe not. I'm not probably using the right words, but it is. It's kind of seen to some extent, if you take the antisense oligonucleotides, how they bind the RNA is H. And siRNA therapeutics, how do they bind? How do they form, the AGO2 and then form the RISC complex? There are definitely certain, structure-based design is included in it. And those are kind of, I think, very well understood now. And the biggest problem has always been in the oligonucleotides is the delivery. Getting to that intracellular space is the biggest challenge. And to address your second question, you were asking about the route of administration, right? The oligonucleotides are mostly delivered by intravenous injection, but some of them are actually injected. The GalNAc siRNA conjugates can be injected subcutaneously as well. Yeah, these are not like dosed directly into your shoulder. But under the skin, you can deposit like a 1 to 2 ml of the solution of the drug, and then it slowly distributes into the body, and that's how the other drug that I showed, inclisiran, is dosed as well.
Yeah. Okay.
Satish,
thank you.
Yeah. Satish, where are you now? Are you at that? Birmingham. Oh, okay. So you've been there for a long time, right?
Yeah, I'm just crossing 20 years right now here.
Wow, 20 years in the same company. That's pretty unusual these days.
Well, I've been moving from discovery to CMC, and now so I was there in discovery for about 17 years, and then moved into CMC. Now I'm mostly managing the world manufacturing our drugs and in several different companies, and also CMC writings. Yeah,
just to let the students on this forum, Satish and I go way back when we're doing our PhD days with the Dr. Ramarao at ISCT. And, he actually made an interesting point, so just now, I just want to elaborate on that. He said initially he was a discovery scientist. That means he was working on the discovery research of identifying new drugs. And in the United States, a lot of pharmaceutical companies spend, quite a bit of their resources on discovering new medicines. And then he also said, the chemists play a big role in that part. And then he also said CMC, this is the chemistry, manufacturing and controls. That means once you identify a molecule for clinical trials, for the development and the clinical trials, somebody needs to make them in the large quantities, right? That's basically a separate division of chemistry manufacturing. And Satish is actually taking care of that, and, it requires a different skill, understanding the, organic, synthetic, organic chemistry, if it is a small molecule chemistry, in-depth understanding, problem solving, and understanding of some of the regulatory, information. So that's a different career path, in case, if just to help you guys to orient your careers in the future.
Yeah, Dr. Ramana, it's a great talk, and actually, you educated me personally. I appreciate what your contributions here. Yeah, definitely, all the students definitely will get benefited out of it. And so I have a couple of questions. The first question that I have is, I know it's how challenging it is, and to delivering right molecule to the right target. And here we are using antibodies to deliver our medicine to the right target. Is it right? So here to see the pharmacological response, so the oligonucleotide is supposed to be released at the site. Is it right? Or is it going to? So how? What is the mechanism for that?
So if that means if we use the antibody therapeutics, is that what your question is? Yeah. So what happens, right, is when you take an oligonucleotide, conjugate that to this large antibody therapeutic or protein therapeutic, the protein's role or antibody's role here is to bind to the cell surface receptor in the target cell or tissue. Once it binds, it gets internalized, along with the payload, what we call as whatever it's attached to it, right? In this particular case, oligonucleotide. And then it gets into the intracellular trafficking, what we call as the endolysosomal pathway. And it gets into the lysosome, where the protein therapeutic is essentially chopped apart, into small pieces. And now the oligonucleotide is left in the lysosomal compartment with a small piece of the protein, or in some cases, actually the last amino acid of the protein to which you conjugated the oligonucleotide. And then from there, the oligonucleotide with a small linker attached to it, or a small piece of the small peptide or an amino acid attached to it, escapes the lysosome and then gets into the cytoplasm, and then does its whether it is RNase-H mechanism or siRNA mechanism, it does its thing.
Okay, so the next question that I have is what kind of bioanalytical challenges you can face with this approach? I can see a lot of challenges. Is that right? Are you going to measure this exposure with a conjugated adductor? Are you also need to, what is the regulatory requirement? Do you need to look into a payload as well?
Yeah, the regulatory requirements depend on the whether you are in the early stage clinical trials or the late stage clinical trials. We're still in the very early stage clinical trials. We do, when we talk about the bioanalytical, right, we do estimate or evaluate the siRNA concentration in the blood, in the tissue. And then when we look at the siRNA concentration in the blood, we actually look at the whole conjugate assay, where you actually pull down the drug by binding to the antibody, and then evaluate the antibody concentration, and then also separately identify the siRNA or the oligonucleotide concentration, so that you can figure out, are there because the way we conjugate is 1 siRNA molecule per antibody. So essentially 1-to-1 ratio, right? Are we maintaining that? Are we just looking at unconjugated siRNA? So we have assays to define that.
Okay, that's great. Thank you so much.
Mm-hmm.
Hi, Dr. Ramna. Thanks for the good presentation. There are a couple of things that we need to take home from these presentations. One of the objective is we are matching students and scholars to mentors. So these presentations will help scholars to basically identify which mentors' research interests are aligning with their interests, so that they can choose the mentor. So my request to students is when you listen a presentation, see if you are interested to pick the mentor, so that offline 1-to-1 you can interact with the mentor, and the mentor can guide you for your career success. My question to Ramna is, it appears there is biology, there is a lot of biology involved in this research along with chemistry. So for our PhD students, in order to get into this kind of research, are there any additional trainings or readings or any career path they need to take, really integrate this biology part into the chemistry? So if you have any insights, then that will definitely help students to align and basically plan for those kind of trainings or any additional classes. Over to you, Ramana.
Yeah, definitely, Ravi. I think I was thinking about that as well. I think this is a very good question, just to help the students, right? Whether you are a master's student or a PhD student, back when I was in India, when we were in PhD students or masters, we didn't really have essentially we didn't really have much connection with the rest of the world, because there is no internet, right? But now you really have the power of the internet. You can just go to Google and Google the term oligonucleotide therapeutics. You can figure out who is working on it, what are the challenges. And there are some really excellent videos put on the YouTube by different companies. I said antisense oligonucleotides. I said siRNA. What are these? How do they exactly work? Right? There is so much information, both the visual information, the information that you can that's something that helps you understand the topic very easily. And then there are other things, you can read books, you can follow the scientific journals, to help you understand or give you much better perspective of the oligonucleotide field. In terms of the biology, I think, Ravi, I may not be able to help you very well, but, I may not answer this question very well, because I'm a chemist by training. But when I look at the drug discovery biology, whether it is a small molecule chemistry or large molecule chemistry, the biology part of it in the drug discovery tend to be pretty similar. That means, you look at a particular cell, and then you are modulating the expression of a protein, or, inhibiting, a particular receptor, or, activating a particular receptor. What are the downstream effects, right? For the biology in the drug discovery industry, they tend to be very similar. I think what changes is that, and that was the question Venkat was asking, the bioanalytical, this is the analytical chemistry. As the molecules become more and more complex, their analysis will become more and more complex as well. And so every time chemists come up with the new modalities, the downstream effect is, the bioanalytics analytical group, they struggle. When I say bioanalytical, it's kind of a rudimentary way of understanding that is, you dose this drug to a patient or an animal, what is happening to the drug, right? Understanding that, so that means you collect the different tissues from the animal or blood from a patient and isolate the drug. Are you saying that it's a large molecule, it is still intact? And how do you develop the assays to understand that? These are the assays that the bioanalytical group develop, and then as I said, new molecules, it becomes fairly challenging.
Hello, Ramana. Hi, this is Prasanna. Very nice presentation on this antisense oligonucleotides and targeting into the cells. Actually, I worked a lot on this antisense platform in my early career. After I finished my PhD in peptides, and then I moved into oligonucleotides, and then later on back into proteins again, and of course now into Rex CMC field. So there's so much transition happen for sure. I know definitely for the students, there is no specific path that they need to be on. I mean, they can keep on transitioning as they go. So that is one thing. And definitely, I mean, in the beginning, as I said, I was working a lot on this antisense platform and stabilizing the oligonucleotides, of course, with the thio, phosphatides. And then I worked mostly on the locked nucleic acids too. What was it? Enzon Pharmaceuticals, and that the pegylation was another, the big thing of stabilizing this or like improving the circulation of the oligonucleotides, right? That was another thing that I was talking about when I was giving the talk. But it's very nice to see that Avidity is working on this DMD, and still there is no cure for it. And I wish, the research and the programs go into next levels and then get the success. And currently you said your products are still in the preclinical stage, or they're into clinic already?
Not yet. We are hoping. We're hoping to get into the clinic before the end of the year.
Okay. Yeah.
Yeah. Yep. Thank you for your wishes, though. I am aware. So you used to be at the UT Dallas, right? If I understand.
No. Actually, no. I did my PhD at Rutgers University, New Jersey, in peptide chemistry, and then I'm in New Jersey all the time, actually.
Oh, I see.
So my research was most like the first initial 10 years was in this oligonucleotides, siRNA, and all that stuff, and then I moved focusing on proteins mainly. And to Ravi's point, I mean, I was like with a master's physics chemistry background, coming into peptide chemistry, and then after that working on this antisense stuff. Even though my major was organic chemistry, and getting into a lot of this biology also. Initially it's tough, but as my PhD program, we used to have a course, a interdisciplinary course with nucleotides, peptides, and the carbohydrates, something like that. So that course kind of covers all the basics, especially the structural part of it, and then of course those bioanalytical things, analyzing different types of molecules, different kinds of techniques and things like that. So those kind of courses also will help the students in terms of learning it as a basic point of view, not just going deep dive into the biology, but at least to understand some concepts. Because we as organic chemists, I know that in my master's or early when I was doing PhD, mostly the molecules and structures and those mechanisms, how to go to this one structure to the next structure, what kind of mainly just the focus is all the time in that, chemical reactions and stuff like that. But at the same time, if we learn the other, those biological molecules, how they work and what kind of structures they have, what kind of mechanism. So I think that kind of courses or some curriculum into the chemistry program will be also useful for the students.
Yeah, I think that's a very good point.
Yeah.
Yeah, I may add a point here. Actually, you know, I agree with both Dr. Ramnagar and Dr. Nagar what they said about the question raised by Ravi Garu. See, I think, you know, even though the chemistry is a fundamental part of the biology, the biologics are becoming more and more prominent nowadays in the drug discovery and drugs, as well as not only the therapeutics, but also diagnostics, right? So what I would see is 2 points. One is, I mean, for a chemistry student to learn a bit more biology, to understand more biology, get into a little bit more biology, rather than sticking to pure chemistry. The 2 points that I do think is, one is on the advisor side during the PhD time, who initiates more interdisciplinary collaborations with the other biology partners, biology advisors, number one. Number two is students themselves proactively looking forward to learn new things, read, understand, learn new things in the biology. I think this is very important. Nowadays, interdisciplinary science, one single skill set is not quite sufficient at the moment in this world right now. I think multidisciplinary skill set is quite important to grow as an excellent scientist. I think that's what I think. These 2 points very important. You know, proactively in the sense, students need to go into a biology lab, your collaborator's lab, and try to watch, try to observe. If you have a chance, go and try to do an experiment. In fact, that's how you would learn different techniques, and you will gain an understanding and expertise on the different techniques as well. That's what I would like to say.
Yeah, very good point, Mahinder. I totally agree. I think, but you know, the information is out there. There's so much information. There are so many ways of learning. As Professor Reddy Garu mentioned, Mahinder mentioned. If you're interested, go talk to your colleagues in the biology department and then really see what experiments they're doing and how can you collaborate with them. I think sometimes if you collaborate with them, you can actually learn a lot more, and that gives you some kind of a reason for you to learn as well. And just to give you an example, during my PhD time, I worked on a synthesis of an alkali that has 30 synthetic steps in one sequence. This is 30. It was like a back-breaking hard work for almost 5 years in Dr. Ramrao's lab. At the end, I made the molecule, but what was the point? I was looking at the NMR tube with like a couple of milligrams of the product. I really didn't know why I struggled so much, made this molecule. What was the point? And I understood that only when I came to the US and especially at this, SMU, when I started working on the antibiotic therapeutics, that Professor John Bayanak was working on the beta-lactamase inhibitors. So I walked in with the chemistry skills, but he introduced me to the medicinal chemistry, the biology part. He taught me some of the biology, gave me some, okay, this is what a beta-lactamase enzyme is. This is how it inactivates the beta-lactam therapeutics. How are we going about it to change that, the enzyme structure, the mechanism of action of enzymes, some of the enzymatic reactions that we do to test the compounds that we made in the lab. It was an eye-opener, but it is a slow learning. It took me a few years to understand the biology part side of the drug discovery. And then I think that's where, once you understand that, right, you really start appreciating the value of chemistry. You really enjoy it as well, because you make a molecule and then someone else will test it. You understand, okay, what has happened, why you expect certain result, but this result hasn't, you did not see that result. So what changes you can make, in the molecule to achieve the therapeutic goal that you are seeking. It becomes pretty interesting.
Yeah, I agree with Dr. Ramana and Dr. Mahinder. Actually, by training, I'm a medicinal chemist, and right now I'm part of the DMPK group where I study different molecules in the drug development, where we try to understand the PK, DMPK properties of all the different small molecules and even proteins. And so what I would like to say is that actually chemistry is a very, very critical component. By knowing the chemistry, you can do anything else. You can learn any other things. And whatever you do, you need to understand the molecule. You need to understand the physiological properties. Everything is linked to the chemistry, and I think chemistry is a base.
It's the fundamental subject of any science. I mean, everything is built upon the molecules, elements that belongs to chemistry, right?
Life begins with chemistry, right? So
exactly.
So we expect at least a couple of questions from students.
Yes, students and scholars. Please introduce yourself, unmute yourself, and ask the questions.
So if there are no questions, shall we close the session with remarks from HOD? Professor Ramesh?
Yeah. Ravi, are you, my voice is audible?
Yes.
Okay. So good evening, everybody, and particularly Ramana sir for giving very good lecture and informative. I hope this time many people have joined from our department. Ravi, is it right?
Yes, I think we usually get some statistics from Dr. Padmasri about how many students and scholars joined later. But yes, that's a very good turnout. Wonderful.
Yeah. Roughly at the beginning, they are roughly around 90, 95, I observed.
Right.
And afterwards, due to some net problem, they left the program. I think so. So whatever it may be.
Unmute.
So, 1 minute, sir.
Ravi, it looks like we lost him.
Yeah, looks like so.
Maybe he's disconnected.
I think there are no other questions. Maybe we can have a vote of thanks from Ms. Wani.
Ravi, one quick thing that I would like to say before the conclusion. So this is to the students and scholars. So what I have seen in the US is many universities have this career guidance, unit to the students, for the students. A career guidance department, a few people dedicated to guide the students on their career, what to do, how to build their skill set, and how to write their resume, things like that. I think this program, we would like to help our students, the Osmania University Chemistry Department students, through this program, which I don't think any university in India or I don't see any place has this, but we have initiated it. I would request all the students and scholars to utilize this program, participate actively. If 1 or 2 students benefits out of this program, we will be very, very happy. We will continue doing this thing, but I request the students and scholars to utilize it. You will see the benefit, and you will feel certainly you'll feel the difference once you have got this benefit. That's what I would like to say, Ravi. Thank you.
Yeah, that brings up another question to me. I would like to ask all the scientists working in US, you all started at one point in your career getting into US PDF. So can you just advise to students how to get PDF?
Sorry?
What are the steps
Ravi.
involved? Yeah,
Ravi?
go ahead. Umesh, please go ahead.
Yeah, some net problem was there. So
Yes.
I would like to give my remarks, and today lecture, it went on very well. And particularly, I'm very much thankful to Ravi Prakash, who has taken the initiative for all these programs, and Hari Padma Sri Madam and Leelavati Madam, and my colleagues in the Department of Chemistry. And I hope the strength may increase next week also. So thank you very much, Ramana Garu, for giving a wonderful lecture and very informative. And I hope we may get some more lectures in the coming weeks. So thank you very much. It is already late. Thank you very much. Thank you one and all.
Yeah. Thank you.
Thank you.
Professor Umesh. Yeah, just to take a quick 5 minutes to I mean, we can take it in any other session also. That's not a problem. But think about this. Give some pointers to students about how to get the PDF. What are the prerequisites to get there, and how to basically what are the are there any websites, are there any groups that they need to pursue, and anything related to that. If you have any information, please share that in various next sessions or offline in the group, so that will be appreciated. And now I request Miss Vani to give the vote of thanks.
Good evening, everyone. A very good evening to our most valued and worthy guest, Dr. Ramana Garu, respected mentors, our department HOD, Dr. Umesh sir, BOS chairman, Dr. Leelavati ma'am, Dr. Padmashri ma'am, and all my research colleagues and MSc students. I am Vani, research scholar working under Dr. M. Vasavi ma'am. Would like to propose vote of thanks on behalf of our chemistry family here. Sir, it was very interesting, informational, and happy to know about oligonucleotide therapeutics. Starting from small molecules, antibody drugs, oligonucleotides, biologics, we came to know about pills that we take, the difference between the pills that we take and new class of drugs, oligonucleotides. We are happy to learn about ASO drugs, short interfering RNA, and the mechanism of action involving in that, inhibiting the molecules and coming up with the new therapeutics and all. Sir, it was the question in my mind even that why there are not many drugs. It is a small, like it's a information that I came to know that phosphorus linkage is not so stable. From your point, I'm happy to know that. And it was interesting when we came to know that how chemists come to rescue in changing the or in modification and modifications made by the chemistry people and playing with the substituents on the moiety, on sugar moiety to change and then and involving in the process and all. And it was motivating with the statement said by you, sir, that to look at the problem and change the molecules so that they become the better drugs. And it was happy to hear about Dr. Muthaiya Manoharan, sir, who was involved in the mechanism how ligands help to increase the intercellular delivery. And it was very interesting to know about the work done on antibody oligonucleotide technology and how oligonucleotides are getting delivered on their promise. Thank you so much, sir, for taking time and mentoring us, educating us, and making us learn about the oligonucleotide therapeutics. Thank you, Dr. Ramana Garu. And we all know that these programs will not or cannot happen overnight. It requires planning and a bird's eye view for details. We have been fortunate enough to be backed up by a team of very motivated and dedicated mentors here who are taking time and for initiating to start this group to help out, inspire, motivate research scholars and MSc students. I thank each and every mentor by name for your time, taking time here, and for your responsible role here, ma'am, and madams and sirs. At last, I thank all the participants for involving in this program and making it success. Thank you all.
Thank you, Ani.
Thank you, sir.
Thank you, Ani. Thank you all, the Department of Chemistry, staff, faculty, students for joining. And please ask your friends to join from next session. They are very useful to your career. All the best to you, and good day for US participants, and good evening for participants from India. We are concluding now. Padmasri, Dr. Padmasri, are you there?
Yes, sir. She has some issue, it seems. So
yes.
She's the message. So she's unable. She's able to hear all our conversation, but she's unable to. We are unable to hear her.
Yeah, probably we need to close the YouTube live also.
Yes. Yes, sir. Once we close this one, we automatically close it. Thank you very much, sir. Once again, Ramana garu.
Yeah, no problem. Okay, bye. Namaste.
Bye. Bye, everyone.